Hello everybody, my name is Vid Jan and today I will present the topic expression of sodium-potassium ATPase, isoenzymes and myokines in cultured human myotubes innervated by rat spinal cord explants. So in the beginning I will just do a short introduction around the subject what sodium-potassium ATPase actually is. Sodium-potassium ATPase is a heterodimeric alpha-beta ion pump and P-type ATPase, which maintains transmembrane ion gradients and is essential for skeletal muscle excitability and contractility. NKA consists of catalytic alpha subunit and glycoprotein beta subunit. In human skeletal muscles, alpha-1, alpha-2, and alpha-3 subunits can be found. Heterodimers from different alpha and beta isoforms are different in their kinetic characteristics such as enzyme and transport activity and their affinity for sodium, potassium, and ATP. Alpha-2 beta heterodimers are predominantly active when the muscle contracts and have a more important role in muscle contractions. Denervation reduces the NKA content in skeletal muscles, while reinnervation increases it. So what were the aims of our study? Well, one was to examine whether myotubes innervated by motor neurons from the embryonic rat spinal cord explants expressed higher levels and or a different pattern of NKA subunits, alpha and beta, compared with the neural cultured myotubes. The second one was to explore how the innervation affects the auxiliary regulatory subunits of NKA called FXOAD1 or F-phospholamine and FXOAD5 or disadherin. The third aim was to examine how innervation affects the expression of myokines which are regulated by contractions. So what were the methods used in our study? Normally, skeletal muscle research in vitro in laboratory is researched on aneural non-innervated mononuclear myoblasts or CD56 positive cells. This can be then differentiated into multinucleated myotubes, but these are not innervated, they do not contract and they normally do not express a striated pattern. In our study, we used rat spinal cord embryonic explants to innervate our human skeletal muscle cells. These differentiated further and in 7 to 10 days started to contract in our culture. So what do our results show? We have shown that myotubes innervated by rat spinal cord explants started to contract within 7 to 10 days and had higher mRNA levels of myokines such as interleukin 6, interleukin 7, interleukin 8 and interleukin 15, while myokines such as musclin, cathepsin B, meteorin-like protein and SPARC were unchanged after 10 to 11 days of co-culturing, while in 21 days of co-culturing, all myokines remained unchanged. Afterwards, we looked at the expression of NKA subunits and FXOADs. On mRNA level, we did not observe any changes in expression of NKA subunits, both alpha and beta subunits. We did not see any change in the mRNA expression of FXOAD1. We did see a slight increase in mRNA expression of FXOAD5, but conversely on protein level we saw a significant increase in protein content of NKA alpha-1, alpha-2 and FXOAD1, but FXOAD5 remained unchanged. We also observed an increase in phosphorylation of FXOAD1, which points to the increase in activity of FXOAD1, which leads to an increase in NKA activity. Due to discrepancies in the results between mRNA levels and protein levels, we also used non-specific primers to evaluate whether there might be some contamination from rat tissue from the neural component. but we did not observe any changes in the mRNA expression with non-specific and specific primers, which points that the differences on protein level are not due to contamination from rat neural component. Afterwards, we also used antagonists of neuromuscular transmissions on the expression of NKA and FXYD proteins, and we did not see any changes in mRNA and levels of NKA and FXYDs with either using bungarotoxin or D-tubocurarin, which are both antagonists of neuromuscular transmission. So, to conclude, our results show that gene expression of NKA isoenzymes in cultured human myotubes does not depend on innervation. We also suggest that innervation and contractile activity in vitro promote myokine expression acutely but not chronically. And in our study, at least alpha-bungarotoxin or tubocurarin did not alter expression of NKA subunits FXYD1 and FXYD5 in innervated myotubes. Thank you all for your undivided attention.